So thank you very much for the organisers for inviting me to speak. Today I want to share with you some work from my postdoc fellowship in Oxford, which sought to explore how individual variability in sensory perception relates to individual variability in sensory maps in the brain and the neurochemicals which underlie these. So I'm sure this is a figure that's familiar to many, if not most of you. Primary somatosensory cortex in the brain exhibits a very characteristic topography along the postcentral gyrus, with a progression from the foot most medial through to the face and the abdomen more laterally. And between these 2 points, we see a really exquisite representation of individual body parts down to the level of the individual fingers and the hand. And it's this finger and hand region that I'm particularly interested in. It's a region which is massively overrepresented in primary somatosensory cortex due to its dense peripheral innervation, and is therefore an ideal model or target region in which to investigate how perception maps back to the organization of the cortex. So various groups around the world, including ourselves in Oxford, have shown recently that it's possible to map the fine-grained features of the somatosensory cortex, in this case the individual fingers, using 7 Tesla fMRI in individual participants. So this is an example of a finger map acquired from about 8 or 9 minutes of fMRI data from one participant in the scanner. And having produced these kind of maps and run this kind of protocol over a number of different participants, here's just an example of 3 in 3 different rows. What's really quite striking is that while within participant, if you look across the different time points here, these maps look very consistent, when we start looking between different participants, there's a striking degree of variability in the relative position and organization of these digit representations in somatosensory cortex. I know as neuroscientists, we all very much like maps. Maps are very intuitive and conceptually easy an easy way of understanding how the brain might be organized. But as someone who produced a number of different maps from fMRI data, after a while I started to wonder, what do these maps really mean? Is this individual variability that we observe in these maps actually meaningful itself? So in one direction, I guess the question is, what's underlying this individual variability in these cortical maps? We know that the inhibitory neurochemical GABA is very important for maintaining selective activation of the cortex, which is essential to producing these very exquisite and detailed maps in the brain. So I guess the question is, to what extent is the variability we see in these maps underlied by variability in inhibitory GABAergic signaling in some sensory cortices? And in the opposite direction, to what extent is the variability that we see in these cortical maps reflective or translating into differences in terms of perception, in this case, tactile perception? So in order to address these questions, we use somatosensory cortex as a model system to understand how the variability in these maps is related to both neurochemicals and tactile perception. So we use the fMRI paradigm I've already outlined very briefly at 7T and combine this with MR spectroscopy at 7T to quantify neurochemicals, specifically inhibitory GABA, and also a more traditional tactile psychophysics experiment outside of the scanner to quantify tactile acuity as a measure of perceptual abilities. So we essentially have 3 different variables here which we're trying to relate at very different scales, moving from the neurochemical to the cortical and then through to something more behavioral and perceptual. In terms of the spectroscopy, we used a sequence at 7T, a semi-laser sequence, which effectively acquires 64 spectra, which we average together and then fit using an LC model to quantify different neurochemicals in the brain, so to fit a concentration of that neurochemical. It's important to note that the voxel that we used is 2 centimeters by 2 centimeters by 2 centimeters. So in terms of spectroscopy, I can assure you that's quite a small voxel, but obviously compared to some of the other imaging modalities that we're familiar with, this is quite a considerable chunk of the cortex that we're imaging. So we get a single spectra for that, locating the voxel over the anatomical hand knob, so a pretty consistent anatomical marker of the location of the hand and finger representations in the brain. And then in terms of the psychophysics, very briefly, we use a temporal order judgment task. So this involves applying vibrotactile stimuli to the index and middle fingers in quick succession and asking participants to judge which stimulus came first. By using a variety of interstimulus intervals, it's possible to fit a psychometric function and then derive a measure of perceptual ability in terms of sensory function in the hand. So again, to draw you back to my favorite figure, what we're trying to do here is relate these neurochemical signals and cortical signals, so a signal of the selectivity that we see in those finger maps using the fMRI, through to the behavioral and perceptual domains. I need to go backwards. How do I go backwards? There we go. So what we see very strikingly is a very strong correlation between the inhibitory GABAergic signal that we get from somatosensory cortex, so at the neurochemical level, with the degree of cortical tuning that we see from those fMRI maps. So in other words, the higher degree of GABAergic tone that we see in somatosensory cortex, the more selective the tuning we see in primary somatosensory cortex from the fMRI. And inversely, the lower the level of GABA that we see in somatosensory cortex, the broader or the more generalized the activity we see from the fMRI data. Presenting the same graph again to you on the left, just comparing this with a control on the right, this is the equivalent correlation using excitatory glutamate quantified from the MRS as well. So you can see that there's a degree of neurochemical specificity in the relationship that we're observing. This seems to be specific to an inhibitory neurochemical in somatosensory cortex. And we've since replicated this result in an independent cohort of 11 participants and shown anatomical specificity in this relationship by acquiring a control voxel in primary visual cortex, where the GABAergic concentrations that we measure do not relate to the cortical tuning that we observe in somatosensory cortex. So this is clearly an inhibitory phenomenon, but also an inhibitory phenomenon specific to neurochemicals in the somatosensory region. And then think about the GABA and their perceptual acuity. So again, we see a strong relationship here. It's important to note that perceptual acuity in this case is measured such that lower numbers mean better performance. So in this case, the higher the degree of GABAergic inhibition we see in S1, the greater the performance in terms of perceptual acuity in the two fingers that we've measured. And again, for consistency, just to present the same correlation with glutamate, this doesn't seem to be a relationship which generalizes across to excitatory neurochemicals. And again, it replicates in an independent cohort and also shows this anatomical specificity. So GABA in V1 doesn't relate to this kind of perceptual acuity that we're measuring. And then finally, thinking about the perceptual acuity and the tuning, both in the original cohort and the replication cohort, we show that the higher degree of the higher the degree of perceptual acuity, the greater the cortical tuning we observe. In other words, if the cortical tuning is very specific, so if we see a very punctate or very finely tuned activation from the fMRI, this is related to enhanced performance or greater performance in terms of tactile abilities. So basically what we have here is a very strong relationship between three very disparate multimodal measures, which are providing information about very different features, both in the neurochemical, the cortical, and through to the behavior. In terms of hypotheses, there's an obvious hierarchy to these kind of different variables. So we would expect that it's the inhibitory GABA which is affecting the cortical tuning that we're measuring with the fMRI, and then it's the cortical tuning we're measuring from the fMRI which is going on to affect the perceptual acuity that we're measuring. In order to explore this kind of pathway through the data, we fitted a mediation model which basically shows exactly that. So there's a very strong mediating effect ongoing here. So the relationship between the inhibitory GABA that we measure in somatosensory cortex and the perceptual acuity is very strongly mediated via the metric of cortical tuning. So it really does seem that a high degree of cortical GABA is associated with a high degree of cortical tuning, and a high degree of cortical tuning goes on to be associated with enhanced or high degrees of perceptual acuity. So obviously, this is only a cross-sectional study, but what it really provides is a very replicable and firm foundation or framework for understanding what's going on in that link between neurochemicals and perception, which is generalizable not only to the somatosensory model, which we've used here, but potentially into other sensory domains as well. The idea of all these results is to build upon them as a framework for investigating the effects of pharmacological interventions to manipulate the neurochemical level here and observe the effects both on cortical signaling and perception, as well as potentially understanding how these processes may become maladaptive in conditions where dexterity or somatosensory function is known to become dysfunctional, such as in stroke or focal dystonia. So with that, I would like to thank my co-authors for helping me conduct this rather ambitious multimodal study in Oxford, as well as the supervision and funding which has supported my work. And I would invite any questions you might have. Thank you very much.
So I didn't understand very well how you derived the measure of cortical tuning. So I saw those blobs for the different fingers. Is that a measure of how close they are together, or how dispersed they are? Or
It's a very good question. So I think if I click here, I will have a slide. So basically, when participants are performing the task in the scanner, it's not a traditional block task. It's a continuous cycle of movement. So they're moving one finger, and then the next, and then the next, and then the next in a cycle. So we're tracking a wave of activity in the cortex. So what we then do is for each voxel, we're able to fit effectively a tuning curve. So where in that cycle of movement do we see the peak of activation? And that's where we derive those maps and the blobs that you were referring to on the cortex. But what we're also able to do per voxel is start deriving these kind of tuning metrics as well, which we can then start to look at the spread of that tuning function during the cycle of movement as a way of deriving that cortical tuning metric. So it's kind of complementary to, but distinct from the maps that I was showing there.
Okay, thanks.
Thank you. Could you quickly run me through how you quantify the GABA to creatine ratios? Because I would normally expect it to be on the order of like 0.1, because we know that creatine is usually like tenfold higher concentrated. And the dynamic range between subjects is super high. You have values between 0.25 and 1. I would expect that to be on the order of 10% max. So how do you arrive at this?
No, again, it's a very good question. So the reference values you're referring to, if we quantify a pure GABA to creatine ratio, that's exactly what we see on the order of 0.1 to 0.3, exactly spot on that. But obviously, given we're looking at comparing between participants and producing a cross-sectional analysis, what we then do is go on to correct for the amount of grey matter that we see in the MRS voxel. So as well as a correction for creatine, what we're also showing, which I didn't elaborate there, is also a value which is corrected for the percentage of grey matter. So adjusting for that, given that GABA is predominantly observed in grey matter, adjusting for the quantity of grey matter that we're observing there. So that's why the values look slightly off from what you might expect.
Nice work, thanks. I just had a question about your psychophysics. Did you collect reaction times at all? And if so, were there any interesting relationships between the reaction times that you saw and these other measures, especially if you're controlling for performance? Like if you have two participants that have similar performance, did you see differences in reaction time as a function of these other factors?
So we did exclude, there was a constraint on the reaction time of about 2 seconds, and we did exclude participants if obviously they started hitting that maximal reaction time constantly. We didn't observe any obvious correlations between the performance and the reaction time, the accuracy and reaction time, but I didn't actually go back and look at the GABA, so that's something that maybe is worth considering.
Great, thanks.
Thank you very much.
Thank you.